in the last class, I had talked about structure of DNA, RNA, and a bit about Watson and Crick's model of DNA. Today I will start with DNA replication. I will continue with this chapter after Kali Puja and other festivities in the next class. But today let me start with some of the basics. So with this I will start to present my screen. You must bear with me. I am in Calcutta right now, and the internet connections in Calcutta, they are not that good. So there may be, although this connection right now it's going well, but I really cannot trust it. So tell me one thing, is this slide visible? স্লাইডটা দেখতে পাচ্ছ তোমরা?
Yes, sir.
হ্যাঁ স্যার দেখতে পাচ্ছি।
বাহ, ঠিক আছে। So fine. So replication. So what do you mean by replication? The first thing what you is in replication, it means that 1 becomes 2. So it's the exact copy of 1. 1 becomes 2. So that process is known as replication. So and also we know that the molecular basis of inheritance is through this special molecule known as DNA. So this DNA is the special molecule for replication, and all genetic information is stored in this particular DNA, and this is the template for replication. This is the basis for replication. Because of this DNA, we are studying DNA replication. So this is briefly the structure of a DNA molecule. Basically, this is this whole region from here to here. This region, this hand is moving. This is 2 nanometer from here to this one full coil. This region, one full coil. This is 3.4 nanometer. Each base is distance between each base is 0.34 nanometer. There is a groove known as major groove, and on the opposite side, this is this groove, this region. This is the minor groove, and A is opposite to T, and there is basically 2 hydrogen bonds. G is opposite to C, and there is 3 hydrogen bonds. Another thing where from this Watson and Crick's model of DNA that came out was that they proposed that DNA is replicated in a semi-conservative manner. Now I am going to explain what semi-conservative manner is in a few slides. So let's go to the next slide if my computer and internet permits. So the basic structure of DNA, like we talked, we have a sugar phosphate backbone. This is a sugar. This little yellow region over here, this is the sugar. This yellow part, this is the sugar. This is this is the sugar, and this yellow part is the phosphate. This is the sugar phosphate molecule plus base. The base is guanine base is added, and so this becomes a nucleotide. And in a DNA strand, we know that these sugar phosphate backbones are on the outside. They are attached like this from the 5 prime to 3 prime direction, and these are the G, C, A, T. These are the different bases. So many of these units, they join together, polymerize together, and form a polynucleotide or a strand of DNA. So a double-stranded DNA looks like this essentially. You have one from the 5 prime to 3 prime direction. I had explained in the last class the 5 prime carbon phosphate, and on the other side you have the 3 prime phosphate, and it's in an anti-parallel direction. One is from 5 prime to 3 prime is from here to here, and the other is from here to here. So, and that is organized in a right-handed coil. So it's wound in a right-handed coil in a clockwise manner. It's wound, and G is opposite to C. You find that there are 3 hydrogen bonds. A is opposite to T. There are 2 hydrogen bonds. The sugar phosphate backbone is on the outside, and the bases are stacked inside in the inside. From this region to this region, there are 10 nucleotide base pairs. They are paired base pairs, and this is 3.4 nanometer in width, and the distance between this to this is 0.34 nanometer. And this whole region, this part to this part, and that is 2 nanometer. Half 1 nanometer, 1 nanometer, 2 nanometer. So this structure was discovered in 1953 by James Watson and Francis Crick. This is a photo of them. They are basically posing for the photograph. This is the model of DNA which they have constructed, and this is Watson over here, and this is Crick over here. Crick is actually showing as if is posing as if is explaining the structure. So basically, this is a modeling done. It's not a real. Actually, it's not a real life photo. So there are other people like I mentioned, Rosalind Franklin and Maurice Wilkins, especially and Erwin Chargaff's. Their contribution to the structure of DNA is immense and cannot be forgotten. James Watson also can be given credit, but essentially a lot of X-ray diffraction studies by Rosalind Franklin and its interpretation by Maurice Wilkins, and based on Erwin Chargaff's rule that amount of A is equal to the amount of T and amount of G is equal to amount of C in different organisms. This basic structure of DNA was constructed by Watson and Crick. So the structure of DNA, you can see that this is you have a sugar phosphate backbone. This is the phosphate. This is the sugar. This is the base. This is the 5 prime. This is the 5 prime carbon. This is the 5 prime carbon over here. This 5 prime carbon is attached to a phosphate over here, and this is the 3 prime carbon from the last plus. This is attached to the 5 prime. So this is the 5 prime end, and this is the 3 prime end. The OH here, it's the 3 prime end. So this is in a 5 prime to 3 prime direction. So this is one strand of DNA. It is attached to a thymine. This is attached to an adenine, and here this sort of a bond here, it is known as a phosphodiester bond. Of course, remember that ATP or ATP, GTP energy is needed in this. So DNA is composed of 4 nucleotides, each containing adenine, cytosine, thymine or guanine. The amount of A is equal to amount of T, and amount of G equal to amount of C, and the amount of purines equal to amount of pyrimidines. This is the Chargaff's rule. And DNA is a double-stranded with antiparallel strands. One is in this direction, the other comes down in this direction. One is in this direction, the other in this direction, and there is a hydrogen bond between bases are inside, stacked inside. So nucleotides in each strands are linked by a 5 prime to 3 prime phosphodiester bond. So this is a 5 prime, this is the 5 prime, and this is the 3 prime. So it's a phosphodiester bond from a 5 prime to 3 prime phosphodiester bond. Some people call it 3 prime to 5 prime. That doesn't matter in this case, but one 5 prime phosphate, carbon phosphate, is attached to a 3 prime carbon OH, and so this is a phosphodiester bond because oxygen is involved, phosphate is involved, and there are 2 ester bonds, so it's a phosphodiester bonds. Bases on opposite strands. Bases on opposite strands are linked by hydrogen bonding, A with T and G with C. There are 2 hydrogen bonds between A and T and 3 between G and C. And if you can see that there is the distance from here to this region, it is broken down. This region is 3.4 nanometer, and the distance between 2 bases is 0.34 nanometer. I don't know if they have seen, and the whole DNA is 2 nanometer. And this region, this big groove, or this region which is there, this is the major groove Opposite to that, it's the minor groove. Bases are in the inside region as sugar phosphate is on the outside. So with this, the basic principle, base pairing to a template strand. The relation between structure and function is manifest in the double helix. Since the two strands of DNA are complementary to each strand, one strand acts as a template for binding a new strand in DNA replication. This becomes clear in this photo. So this is the parent DNA. This is A, C, T, A, G. This is the parent, and opposite side T, G, A, T, C. This is the parent DNA. During the first step of replication, they unwind and the two strands they separate. Then nucleotides come over here. One of this because it acts as the template for addition of new nucleotides. The same happens on the other side, and like this, two strands of DNA are formed. This type of replication is known as semi-conservative replication. That is, one strand of DNA, it acts as the template for the next strand which is formed. This acts as template for the next strand which is formed. So this there is an experiment of Messelson and Stahl. Possibly I will explain this experiment in the next class because questions may come, but right now, take it from now, that there were three models of replication. One was conservative, one was dispersive, and one was semi-conservative. Maybe I could show you the picture if it's open already here. Considering the slowness of computer and internet connection, I'm not getting confidence. See if it comes. Are you able to see this? Semi-conservative model, conservative model, dispersive model. কেউ একজন আনমিউট করে একটু বলতে পারবে?
স্যার আমরা ভিডিওটা কবে পাবো?
ভিডিওটা আজকেই পাঠিয়ে দেবো।
স্যার
আজকেই পাঠিয়ে দেবো। এটা দেখতে পাচ্ছো?
স্যার
হ্যাঁ।
হ্যালো স্যার।
হ্যাঁ হ্যালো।
হ্যালো।
হ্যাঁ হ্যালো।
ঠিক আছে স্যার।
শুনতে পাচ্ছো?
ঠিক আছে, তাহলে আমি একটু লিভ করছি।
আচ্ছা, ওকে। এই তোমরা কেউ শুনতে পাচ্ছ কিনা আমাকে একটু বলবে?
হ্যাঁ বলছি যে, হ্যাঁ স্যার বলছি, হ্যাঁ হ্যাঁ শুনতে পাচ্ছি স্যার।
আচ্ছা, ওকে, ঠিক আছে, তুই লিভ করো তো।
হ্যাঁ স্যার শুনতে পাচ্ছি, আপনি বলুন।
আচ্ছা, ঠিক আছে। আর এটা দেখতে পাচ্ছ সেমি কনজারভেটিভ মডেল, কনজারভেটিভ মডেল, ডিসপারসিভ মডেল?
ইয়েস স্যার।
এটা দেখতে পাচ্ছ না সেমি কনজারভেটিভ মডেল? তোমার এখন স্ক্রিনে কি দেখাচ্ছে?
স্যার দেখা যাচ্ছে, আপনার ভয়েসটা মাঝে মাঝে একটু কাটে কেটে আসছে।
ভয়েসটা কেটে কেটে আসছে, কিছু করার নেই। আজকে এটা কলকাতায় করছি তো, কলকাতাতে মানে জিনিসটা খুবই বাজে আসে। জানিও না ভিডিওটা ঠিক করে রেকর্ডও হবে কি না ভগবান জানে। এনিওয়ে, তো তো এই স্ক্রিনটা দেখা যাচ্ছে কি সেমি কনজারভেটিভ?
হ্যাঁ স্যার দেখতে পাচ্ছি।
Okay, so semi-conservative model, essentially this is this pink color strand, this is the parental strand. So after the first cycle, what happens? This blue is the new strand which is formed, and this pink is the old strand. So these two strands separate, and each strand acts as a parent for the other strand. So now this blue, this again separates, and the blue forms the new strand, and this is the older strand. So this is semi-conservative. In the conservative model, as you can see, the whole template acts as a template for a new strand. Both the strands are new, and here also both the strands are new. But this is the old strand, as it is the parental strand is over here, and this is the completely the new strand. And here half are new, half are old, half are new, half are old. And in dispersive, it is all mixed. Pink, blue, pink, blue, pink, blue. It is totally, it is mixed. Some part is old, some part is low. Here one whole strand is new, and one whole strand is old. And so this is the semi-conservative model. So now let's go to back to the slide. Just wait for a moment. This slides. This is very slow. My computer is slow. Along with the problem of my. Okay. Internet connection. So after we finish this, DNA replication is semi-conservative. So by this, what do we mean? So this red is the original strand, this purple is the also another original strand. So they separate, and each strand acts as a template for a new strand. A new strand is synthesized on top of the old strand, and this region is known as the replication bubble. So the double-stranded DNA, where you can see that this is the inside the whole genome, the DNA is organized. So little bubbles are formed where the strands unwind, and on top of that a new strand is synthesized. Gradually this opens up completely from in both directions. Replication is bidirectional. It can occur in both directions, and once it occurs, you suddenly find two new strands are formed and a new strand of DNA is formed. So each two-stranded daughter molecule is only half new. One original strand was used as a template to make the new strand. This is how DNA replication takes place. Few facts on the DNA replication. The copying of DNA is remarkable in its speed and accuracy. It involves unwinding the double helix and synthesizing two new strands. More than a dozen enzymes and other proteins participate in DNA replication. The replication of a DNA molecule begins at special sites called origin of replication, where the two strands are separated. So, where the two strands are separated, that is known as the origin of replication. If you go to the last slide, this is the region where they in a DNA there are regions where the strands start to separate, and that region is known as the origin of replication, and that has got a specific sequence. So whenever the enzyme for DNA replication, which is known as the DNA polymerase, comes across this particular sequence, it understands that this is the region where I should start the replication. So there in a long DNA strands, there may be different origin of replications, especially in the case of eukaryotes, which have long DNA organized on chromosomes. Prokaryotes are relatively small, and prokaryotes, another fine thing happens, it's circular. So it's circular, there are plasmid-like structure, a circular structure. So circular also bidirection, that's also has got a new way of doing it. It's straight in the case in chromosomes, but it's circular in bacteria. So more than a dozen enzymes and other proteins participate in DNA replication. This is an interesting part, of course. We will be talking about those enzymes. So what are origins of replication? A eukaryotic chromosome may have hundreds or even thousands of replication origin. Where replication starts, that is known as the origin of replication. So actually it was a single double-stranded DNA. This bubble is formed in many places. They have got many origins of replication, and the region where it is separating, this region, this box over here, this is known as the replication fork. And this is gradually on this template, the other DNA is being synthesized in both directions. So eventually the light blue is the new daughter molecule which is formed, and the deeper blue was the old strand. So this is one strand of DNA, and this is the second strand of DNA which is formed here. So replication essentially begins at specific sites where the two parental strands separate and form replication bubbles. Parental strands are separated and formed a replication bubbles here. Then the bubbles expand laterally as DNA replication proceeds in both directions. So they are proceeding in both directions. Dudikei jacche, both directions. And the new light strand is formed. Eventually the replication bubbles fuse and synthesis of daughter strand is complete. In eukaryotes, DNA replication begins at many sites along the giant DNA molecule of each chromosome. So let us talk about the mechanism of DNA replication. DNA replication is catalyzed by DNA polymerase. The enzyme is known as DNA polymerase, and for this it requires a small strand of RNA known as RNA primer. The enzyme which attaches the primer is known as RNA primase. It synthesizes primer on the DNA strand. DNA polymerase adds nucleotides to the 3' end of the growing strand. So the template strand is in the, kindly note that it is in the 3' to 5' direction, and the direction of the replication is taking place in the 5' to 3' direction. Remember this point. So this is the single stranded region. In this region you find that specifically you find that this pink color is the small RNA primer which attaches by the enzyme known as primase. Then DNA polymerase cannot start. It needs a little primer to which DNA polymerase can add and synthesize. So on top of this strand, DNA polymerase adds DNA nucleotides to primer. So behind this RNA primer, DNA polymerase makes this new DNA. So this is how this is the basic mechanism of how replication takes place. So nucleotides are added by complementary base pairing. Complementary means complementary to A is T, complementary to G is C, and the other way around. So the substrates are deoxyribonucleotide triphosphates, that is DNTPs. It can be D-adenoside triphosphate, D-adenosine triphosphate. Last day, if you remember, it can be D-guanosine triphosphate. All these things, just remember those nomenclatures. So NTP. So this is the template strand which is separated. This is the 3' end OH. Dekho, sugar ta ulto ache. The sugar is in the other way around, and the adenine base is attached over here. So this is the complete whole strand. Opposite to that is you can see that the sugar phosphate is on the outside, the bases are inside. So these have already been formed. Now this is the growing strand. In the growing strand on the 3' end, say opposite to G will come C. So this whole DCTP, there is 3 phosphates. See 3 yellow bubbles, there are 3 phosphates, but here there is only 1 phosphate because 2 have been hydrolyzed out. So this will come and hydrogen bond over here, and because of that enzyme pyrophosphate ion, 2 phosphates will be removed, and this energy will be spent in forming this phosphodiester bond. First this will come over here. This alcohol will attack this sort of phosphoric acid end, and then ester will form, and 2 pyrophosphate, they will be removed out. So just hold on. It seems my mouse is not functioning. In Calcutta, this is the problem, it happens. I have got defective equipments. So you can see that this pyrophosphate moved out. Now this mouse is working again. So let us go to the next slide. The mechanism of DNA replication. DNA synthesis on the leading strand is continuous. So because of certain problems with defective, I will not go ahead. In the next class, I will continue this. So I'm sort of ending my class over here because of equipment problems. But I will sort of send this video. After this basic, I will continue with the mechanism of DNA replication. What is lagging strand? What is leading strand? This I will do it on this Wednesday. Hopefully, I will try to sort out my computer problems. And if required, we can do this on Cisco WebEx, where I don't need to record it externally. Okay? আচ্ছা তোমরা কি এখন শুনতে পাচ্ছো?
হ্যাঁ স্যার।
হ্যাপি কালী পুজো।
হ্যাপি কালী পুজো স্যার।
বুধবার দিন বাকিটা করে দেবো, ঠিক আছে? আজকে কিরকম মানে আটকে আটকে যাচ্ছে, এবার কেলোর কীর্তি হওয়ার আগে আমি সেটাকে সেভ।
হ্যাঁ স্যার, ছুটি দিয়ে দেন।
হ্যাঁ, যে ছুটি দিয়ে দিয়েছি। আমরা বুধবার দিন আবার বসবো 11:30টার সময়। সেম লিঙ্ক, লিঙ্কটা একই থাকবে, আমি রেকারিং করে রেখেছি।
ঠিক আছে স্যার, রেপ্লিকেশন করাবেন তো?
রেপ্লিকেশনটাই বাকিটা কন্টিনিউ করব। আর তোমাকে বলে দিচ্ছি যে দরকার হলে সিসকো ওয়েবএক্স এ করব, এটাতে আরেকটু রিসোর্সটা কম লাগবে।
স্যার একটু না রিপিট করে দেবেন, মানে ভালো করে বুঝতে, মানে কথাগুলো কেমন কাটা কাটা করে আসছিল, নেটওয়ার্কের প্রবলেম হবে।
নেটওয়ার্কের প্রবলেম হোক, আগে পরের দিনও সলভ হবে না জিনিসটা, যতক্ষণ না আসানসোলে যাবো।
ঠিক আছে স্যার,
ঠিক আছে। তো ভিডিওটা পাঠিয়ে দেবো, আশা করি ভিডিওটা ঠিক হয়। দেখি কি হয়।